Yep. I can't find it, so.
Hopefully it's live. If it's not, then, well, we got a problem.
It's live. It's live.
I see it.
It's
Okay.
It's live. It's live. I didn't. Oh. It's fine. It's fine. It's fine. It's fine. It's fine. We're good. Everything is
Are you
all right.
okay?
No.
All right. Okay, same.
Turn the Kahoot volume down a little bit, Ding.
Okay. Is that better?
I have to wait for the stream to catch up to it.
Yay. We love delays.
For those of you who are here watching with us, please click the link for the Kahoot
Yeah,
channel.
the volume's good.
That way
Okay.
there won't be a lag. So don't click next or go until we go, so you can hear the explanations for the stuff. But yeah. Do.
I like the Christmas theme a lot better than the original theme.
Yeah, it's nice.
I can't hear it, but probably it's the same thing from yesterday. Don't worry about that.
Don't even worry about it.
Yep. All right, we're gonna give it like a minute and then we'll start or something like that.
Assuming.
Nice. Yeah, so make sure you go to our description where you can find the notes and that sort for AP Bio, as well as other videos and podcasts that we made for AP Bio. And yeah, it will be a fun ride. All right. Do you guys want to start?
Yeah. All right. Here we go.
Let's go. Yeah. So, do you want to read the problem?
A leaf building up a proton gradient has been removed by an organic solvent. What would most likely be in the solvent?
Right. So, think really carefully about just basically a leaf. So, what does a leaf have? It's not a very complicated question. And what an organic solvent does, it basically just dissolves, materials, or dissolves. In this case, dissolves the leaf. All right, Ding, you need to tell us when time is up, we can.
Time's up.
All right. I'm lagging really bad. Like my Kahoot is just a blank screen. Anyways, so the answer to this is chlorophyll. And so if you think about it, it makes sense because in a leaf, the thing that it has the most is chlorophyll. Due to it being a leaf. I'm not sure how to explain this. Yeah, my Kahoot's just a blank. It's not loading. But yeah, so
that's great.
Yeah, I know, right? So the so the thing you should notice here is just know what an organic solvent does and what is most prevalent in a leaf. The rest of them don't really make that much more sense. But do you guys want to continue?
Yep. All right. Let's move on to the next question. Anabolic pathways yield energy by.
Yeah, this one's a tricky one because it's worded weird. But basically, what it's talking about is how does it.
Time's up.
What? Oh. So explaining what it does is anabolic pathways. They don't yield energy per se, but what this question is kind of implying is basically like, what can it do to produce things that can make energy. Do you know what I mean? Or am I not being very clear?
The question is just worded a bit weirdly, but like, since
Yeah,
anabolic pathway doesn't actually like produce energy and when
right.
like functions, but like it uses up energy to like be stored in the bonds.
Yeah, exactly. So it uses up energy to make bonds to produce more energy. So the bonding of the water and carbon, if you take that apart, then that produces energy. Anabolic versus metabolic, right? Or am I thinking
Catabolic.
of something else? Catabolic, yeah, not metabolic. It's all right. It's all right. It's all right. All right,
Alright,
moving on.
yep. Which mechanism is not involved with the regulation of the citric acid cycle?
All right, so make sure you remember what the citric acid cycle is, where it occurs, and etc., because it does have ties with this question.
Yeah. And a side note, the citric acid cycle is also commonly referred to as the Krebs cycle.
Right.
Yep.
Yeah.
Alright, time's up. So the answer is oxidative phosphorylation.
Yep. Do you guys remember what that is?
Oh, yeah, I got it. The oxidative phosphorylation, you can tell that it's not a mechanism involved in the regulation of the Krebs cycle because it's an entire different process in cellular respiration. It's the final process where the reactants oxygens are actually used in order to produce like water and whatnot, whereas
Right.
the citric acid cycle or the Krebs cycle will use products from glucose to make carbon dioxide instead.
Yep. It's like really helpful to actually look at like the diagram that shows like the circle or like what processes are involved because there's a lot of steps.
Yep.
Alright, shall we move on?
Sure.
Okay.
Wait, yeah, just adding one more. Oh, never mind. You go.
It's the graph question.
Oh, well, that's sad. That's just.
Do you want
sad.
me to like read
All right.
the little pre-graph thing and then read the question?
Yeah.
So I have the picture pulled up.
Sure.
So the graph shows that researchers studied the activity of, I'm going to pronounce all of these wrong, mevalonate kinase, which is an enzyme that may be important for biofuel production. And the graph below shows that the rate of a reaction as the concentration of ATP is increased while the substrate concentration remains constant. The open circles on the graph represent data points collected at 30 degrees Celsius, and the closed circles represent data points collected at 37 degrees Celsius. How is the reaction between enzyme and substrate modified by the different temperatures?
Right. I feel bad for those who can't zoom in on their computer. That's fine.
And you can, on your browser, you should be able to do like the Control+ to zoom into the picture.
Oh, okay.
I don't remember if clicking on it lets you zoom in, but I don't
Yeah.
get it does.
Oh,
I know, but the
time
okay.
is up.
Yeah, time is up.
Yeah.
All right. So the answer is triangle. So if you notice in the graph, you might need to zoom in. If you can't zoom in, well, that's too bad. But the 30 degrees Celsius is the lower one, and the 37 degrees Celsius is the upper one. So if you do remember, if you increase the temperature, it increases the rate of collisions. And if you increase the rate of collisions, that means there would be an increase in the rate of the reaction. The rest of them don't make that much sense. Yeah, so like D is definitely wrong. C, enzymes for correct shape to maximize reaction rate. At 30 degrees Celsius, enzymes are denatured. That's definitely wrong because lower temperatures won't have the enzymes denature. It's higher temperatures. So yeah. And B is wrong because at lower temperatures, there's less less kinetic. Well, yeah, lower temperatures there's less kinetic energy than higher temperatures because the molecules are moving at a slower rate. So like temperature increases the rate of both reactions and just bumping each other in general. So kind of like when you have a fever, right? If one of you want to explain that.
I don't know where you were gonna go with that.
Okay, well, well, it's kind of similar, kind of not similar, but enzymes react faster under higher temperatures. So,
depends.
well, some cases, if it's like extreme temperatures, then it's just gonna denature.
Okay, but like the reason for a fever, if you guys are curious, I don't know why I'm doing this, but it's because like the virus or bacteria that's inside your body also has enzymes. So what your body is doing is basically like dealing damage to both yourself and the bacteria. So like both your enzymes and the bacteria's enzymes are not going to function well. So like it's actually more beneficial this way than like leaving it in a normal temperature. That's why like you kind of feel drowsy when you're having a fever.
Yeah. Yep.
But yeah, higher temperatures
all right.
increases
Do you guys want to
randomness, collision. You kind of learn more about that in chemistry. Check out our chem live streams and podcasts, by the way.
Yep.
All right, next question.
Can we read it? Which of the following molecules would electrons be found after they have been released from the electrons transport chain?
Yeah.
Glucose.
Oh,
sorry.
No. I was just about to explain it. It's fine. You can... Well, time's up.
Well, not yet. Almost.
Oh, well, it is for mine, but it might be lag. It's fine. But what the answer choices? The answer is oxygen because if you remember in electron transport chain, if I'm not wrong, oxygen's the final carrier. I think I'm right.
Yeah.
Hold on. Yeah, electron receptor. That's the word. Okay. And yeah. It's not the others. It's not that I'm not sure how to explain. Mostly you need to know what the electron transport chain looks like. So just keep that in mind.
In some ways, all of the other molecules are kind of too big. And like oxygen is the one that's like actually requiring an electron because if you don't remember, it's actually just one oxygen molecule and not two, which is why like it needs the electron. Right. Carbon dioxide doesn't make sense. Glucose doesn't make sense. That's a huge molecule. Water also doesn't make sense.
Yeah.
All right. Moving on.
In the electron transport chain, there is a no. I was just gonna add in the electron transport chain, there is a production of the water, but it doesn't have to do with this. So, yeah.
Yeah.
Or am I wrong? No. I think I'm right. I'm pretty sure I'm right. Flashback to the past where I thought chemiosmosis was the electron transport chain. We're not going to worry about that.
Moving on.
Moving on.
All right. Next question. So the enzymes that break down polysaccharides can remove monosaccharides only from ends of the molecule, which explains why animals use glucose as glycogen and plants use it as starch.
This is an interesting, tricky question. Yeah, in bio, a lot of the questions are like, if you know there might be like multiple concepts together. If you just know one concept, you can like get the answer. So.
Thumbs up. So for this one, you need to like know the structure.
Yeah.
And like what purpose it serves for like the organism.
Right. And you also need to know like highly branched structures can release more glucose. If I'm not reading that incorrectly, because it probably
Yeah.
Probably I'm doing something wrong. But so D is definitely incorrect because if it's not present in animal cells, I mean, not in plant cells, then that doesn't make sense. Um, C, plant stores starch in roots while animal stores them in muscles and liver. It kind of is correct, but it doesn't tell, like, it doesn't
It
answer
doesn't
the question.
explain why. Yeah.
Yeah, it doesn't answer the
Yeah.
question. And well, B is not really. So it's A. That's
Doesn't
a
really have
great,
that much
great
to do with
explanation.
osmotic pressure or Turgor. No, Turgor. Turgor?
Turgor, something like that. Something like that.
Alright, next question. Which of the following is the function of the endomembrane system?
So remember what the endomembrane is. Y'all remember what the endomembrane is?
Are you asking the stream people or us people?
I'm just asking in general.
Well,
Yeah.
Asking
hopefully we
someone.
do.
All right. The endomembrane system it regulates protein traffic and performs the metabolic functions in cells. So that's the C answer, the yellow circle. That wouldn't be the production of ATP because that's in mitochondria, and the endomembrane system includes like the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, and then the different like lysosomes, vesicles, and vacuoles. So mitochondria isn't included in that.
Right.
And yeah, the production of proteins through mRNA from the nucleus isn't part of like any of those components that I just named, so that wouldn't be it. It's not like a key function of the system. And the production of lipids to restore the membrane bilayer, that's also not really a thing that it deals with.
Yeah, these are a lot of definitions. So,
Yeah,
yep.
I think this is Unit 2, right? Unit 2 is a lot of just memorization.
Yeah, it's Unit 2.
Alright,
Oh,
next
Unit 2 is
question. Unit 2
huge
is a
chunk.
pain.
Yeah.
It is.
Which of the following features is present in prokaryotic plant and animal cells?
Whoa.
So again, more memorization.
Yep. So you can definitely like front off eliminate cell walls and membrane bound organelles because cell wall is something that animal cells don't have. I think plant cells have it, and prokaryotic cells, some of them have it. Remember?
Well, I hope plant
What?
cells have cell wall.
Yeah,
Plant cells
that
and
would be
like
kind of bad.
plant cells and different fungi have the cell walls.
Right. And membrane bound organelles. If you remember, prokaryotic doesn't have membrane bound organelles. And nucleus, ribosomes is the correct answer.
Yeah, prokaryotic cells aren't that like sophisticated.
Yeah, they don't have the
Alright.
membrane-bound organelles or the nucleus.
So the only thing they have in
Alright,
common
alright.
is ribosomes. Yay.
Whoa. Yeah, I mean, it makes sense. Otherwise, they wouldn't be able to, like, live because proteins. But
Yeah.
proteins and other stuff that ribosomes make.
Alright, next question. Which of the following do not require transport proteins to pass through the membrane? We're given water. I don't remember my chemistry. I think that's nitrate.
That is nitrate, yeah.
Okay, carbon dioxide and glucose.
Or you can eliminate one of them immediately.
I wonder which one.
I'm not sure, man.
Yeah. I don't think any of us know.
Not at all.
Alright, time is up. So I wonder which one we can eliminate. Glucose. It's a little bit big, you know?
Whoa.
Just a little bit.
Just a bit.
Just in comparison, just a bit.
Yeah.
So focus on what you do know. But so only certain things can pass through the phospholipid membrane because of the phospholipid bilayer with the heads and tails. So only small and Wait, hold on.
Non-polar.
Okay, yeah, nonpolar. I'm so afraid of saying the wrong thing, man. So small things and nonpolar can pass through. So glucose is too big. If you remember water has its specific protein channel, so that's not it. So if
And
you
it also is a polar molecule.
oh yeah, that too. And whatever NO3 is, I forgot what you guys said versus
Nitrate.
CO2.
Nitrate.
Nitrate, right?
Notice how it
Versus
has a
CO2.
charge.
Yeah.
That's why it
Yeah.
can't pass through the
It's
membrane.
polar.
Yep.
Yeah.
And really quick, before we move on, the reason those polar molecules can't pass through the membrane is because the inner portion of it, the lipid tails, those are all nonpolar, so they can't interact well with polar molecules.
Yep.
Yeah. Yeah, bio isn't hard, but if you're not clear on what stuff is, you'll be completely lost and disoriented and driven insane. So
It's mainly memorization, honestly.
Yeah.
a lot of that. All right, moving on. We're barely on the 10th question.
Speed run.
What happens if you place a red blood cell in a hypotonic solution? So that'll be like a pure water solution.
All right. Yeah, it's D, guys. Anything can happen. It's up to fate.
Well, for sure. I think
Yeah.
that's obvious.
It's obvious.
Who
All right,
Mhm.
knows what can
tell us
happen.
whenever.
Alright, time's
know,
up.
right?
Yeah.
Yep. Okay, so this should be easy to picture if you're clear on hypotonic, hypotonic. I didn't say the same word. Hypotonic, hypertonic, and isotonic.
Big brain time.
So in this case, A is the right answer because if a cell's placed in a hypotonic solution, then the cell will burst.
Yeah, and the reason it's going to burst is because the water will move down its concentration gradient. So it'll move from an area of high concentration to an area of low concentration. And since the red blood cell is being placed in a pure water solution, a lot of the water that's in that solution is going to be trying to flow into the blood cell all at once. And that'll cause the cell to overinflate and burst.
Yep.
In contrast, when you have like a hypertonic solution where like concentration is much higher outside, the water is like gonna come out of the cell, and so the cell will shrivel
Right.
up.
Right. So that's why you shouldn't swim in the Great Salt Lakes too long because you'll shrivel up.
Fun.
I mean, technically you shrivel up like anywhere, like even just taking a bath because
Yeah, and like
there's more water on the
finger your
outside.
the finger on your skin.
The
No, the skin
finger
on your finger.
Yeah, you get like raisin fingers, kind of like.
Yeah.
Yeah.
You're losing water, so just for you to know.
Yep. All right. Next question.
Whoa.
Which of the following is an accurate representation of phagocytosis?
Oh.
Yeah,
So
the more
remember what
memorization of terms.
phagocytosis.
This
Remember
one like,
what
is easier because
phagocytosis.
there's like pino, phago, so it makes sense. Well, at least to me.
Yeah. So remember pintocytosis, phagocytosis, and there's endocytosis, right? But I don't know if that. Am I talking about different things?
I think endocytosis and exocytosis are two different things from the phagocytosis stuff.
All right.
I think.
Time's up. So
Okay.
the only one that makes sense for phagocytosis, which is like engulfing large objects basically, is an amoeba engulfing a bacteria using its membrane to surround it. A doesn't
Right.
make sense because it's enclosing upon smaller molecules. That would be pinocytosis. Receptor on the membrane detects a particle of or ligand. That would be receptor mediated. And vesicle inside the cell fuses with the membrane. That's the opposite of phago and pinocytosis.
Okay, yeah. And clarifying, Nico, you're partially right, and I was partially right. Anyways, so the whole overarching term for it's endocytosis. And within that, there's phagocytosis, pindocytosis, and receptor-mediated endocytosis. So those are those three terms. So just wanted to point that out.
Yep. All right. Next question.
Yep.
Which of the following is inaccurate about compartmentalization?
Everything. All right. So yeah,
I
remember what? No, you can
Sorry.
go.
I was gonna say, I think you're right. Everything
Yeah,
is inaccurate.
everything is inaccurate. All right. So yeah. So remember what compartmentalization does. That's basically it.
Oh, there is a grammar mistake.
We
Uh-oh.
don't worry about that.
Good thing we're not AP Lang, right?
Don't worry about it.
So time's up. It is the one with the grammar mistake. Compartmentalization decreases the efficiency of cells. That's
Right.
not right, because
It's not right.
compartmentalization actually
Yeah.
increases the efficiency because it splits up the cell into different regions. So, like, as we talked about before, enzymes function better in different conditions. So, like, it can change like the acidity and that
Right.
sort to allow the enzyme to function at its peak.
Yeah.
Basically.
Yeah, it has better optimization. Another thing to note though, like for example, like in prokaryotes, I did not call them prototypes. In prokaryotes, when they're doing transcription, translation, etc., since they don't have those membrane-bound organelles, I can definitely talk
You're
today.
good.
They don't have that compartmentalization, and although it is faster, it's not efficient in other cases, in most cases, because of what Dean was just explaining of different enzymes in different conditions. For example, like the membrane organelle that digests the waste products, lysosomes. That's the name. And then other ones like peroxisomes, I think. Those are the ones in your kidneys, if I'm not wrong.
Yeah.
Yep. Okay.
So endosymbiosis is common explanation for the origin of compartmentalization. That is correct. If you think about like the mitochondria, like how its genetics is different, that sort. They're not seen in prokaryotic cells because prokaryotic cells are too small, and it does function more efficiently. It allows the organelles to function more efficiently.
Rep.
All right, next
Rep.
question.
Yep.
Explain what an organic compound is.
Unit 1.
This one's definition too. Whoa, unit 1.
Yay.
Chemistry, my least favorite unit. Whoa. All right.
You all chemistry
So-
haters.
No one likes chemistry.
Yeah, I don't know who does.
Yeah.
Nico's definitely not a chem tutor.
Yeah, we don't worry about that. We don't worry.
All right,
Yep.
time's up.
Okay, so yeah, this one's definitions again. But the answer is A. Molecules have carbon and hydrogen in them with some other molecules. And yeah.
Yep.
Just the main thing you need to know is that all organic compounds will need to have both carbon and hydrogen. They can't have like only one. They need both of them.
Yep.
Mhm.
Again, more
All right.
memorization. Basically what bio is. All right, next question.
Yep. All right.
What happens when an atom absorbs energy and the electron moves to a higher energy level? Whoa.
It becomes a
Wow.
god. All right. Wow. All right. So yeah, so this one's definitions again.
Another chem question.
Oh, how much I wish how much I wish it was D. It becomes
Or
overwhelmed and stops functioning.
You stressed out.
All right. So the answer for this one would be B, because the electrons go into a more excited state when it goes to a higher plane or higher energy level.
Yep. That's how
So, yep.
photosynthesis works.
Wow.
So obviously,
Well, yeah, part
A
of
is
how
wrong.
photosynthesis works, but
well, yeah, parts of it, like how the chlorophylls
Yeah.
work. Because obviously A is wrong. Something happens. The electron moves into a ground state. That'll be after when like the electron releases back like the energy that absorbed from lights or whatever. And the atom becomes overwhelmed by the energy and does not function anymore. well, then we would all be dead. So I don't think that's right.
I
That's a bit of a problem.
the next question is we got a typo there, but it's fine.
Don't even worry about it. All right, next question. Which of the following is inaccurate about the characteristics of water?
Water is polar.
Water.
Water is polar, but we have a problem. We got two inaccurate ones here,
Well,
but
you
we
can
didn't
take
put
multiple
multiple
answers.
choice.
Did
No,
we
we
make it
didn't.
multiple choice?
I don't think we did.
Oh,
It's all right. It's
Oh,
all right.
that's my
Don't
fault.
pick D, guys. It's A, B, or C.
Well, D wouldn't be right anyways, because it's saying which of the following is inaccurate about water, and D is that water isn't. Oh, wait a
Wait.
minute. Never mind.
Wait.
Yeah,
I read
water.
the question wrong. my
Wait,
bad.
hold on. This is tricking my brain. It's all right. Just know water is polar. That's all
Water
we
is
need to
polar.
get from this.
Yeah. So water is a universal solvent. You probably heard of that a lot of times, from somewhere at least, hopefully, because it dissolves most things because it's polar. You'll learn more about that in chemistry again. I hate
Yeah,
chem.
water is polar.
Right.
Water allows for
Yeah,
capillary action.
and yep.
Yeah, that's how
trees.
plants work. Yeah. And
Mhm.
water is not polar.
So water's also universal solvent, by the way, if you don't know. That's why you put things in water if you have stains. Well, technically you don't. Depends on what kind of stain it is, but we don't worry about it. Sometimes water doesn't work on stains, but
Like
it's fine.
I
It's
mean, with
fine.
some
It's fine.
detergent, it does.
Yeah, but that has to do with something else. We'll
Yeah.
get to that in the future.
Hopefully.
So yeah, so the answer is B. Water has low cohesion properties, and we should get a move on because we're on question 16.
Yeah.
Okay. Next question.
Run.
Which one is not a function of proteins? Ooh, I wonder what a protein is.
Wow.
I'm not sure, man. It's definitely Tide Pods. Just kidding.
Nice.
Don't eat Tide
Obviously.
Pods, guys.
It's proteins are definitely not polypeptide chains of amino acids.
They're not important at all.
Nope. Yeah. Oh, and if you guys don't remember protein folding, that's interesting. You guys should remember it. Re-remember it. I'm talking to the audience, not you
Remember
guys.
it.
All right, time
Yes,
is up.
re-remember. Okay,
Alright.
so we know it acts as hormones. We know it's membrane channel because membrane channel protein. But we also know that it acts as enzymes. So if you know what a nucleotide is, you know it's a nucleic acid, not a protein. So that's
Kinda in the name. Yep,
all
just
right.
more
Moving on.
memorization. Alright, moving on. Which carbohydrate has the simplest sugar and serves as an energy source for cells?
Definitely is none.
Yeah, none of
Yeah,
them.
none.
None is the right answer.
What's a sugar?
I'm not sure, man.
What's a carbohydrate?
No, we don't know. What's energy? All right, so this is also definitions. But if you know your root words, you can find which one this is. So there's mono, di, and poly. So just think about it in your head: mono, monocycle; di, dicycle; tricycle. All right,
We don't
I'll
really
put
have
that
a try,
aside.
but okay.
It's fine. Yeah, it's poly. It's up. It goes up to di, and then after that's poly. But
Yeah.
it's fine. I just
So
wanted to put
mono
that
is 1, di is
pun
2, and
in.
then poly is just multiple.
Yep.
Yep.
All right. So moving on to
Moving on.
the
Yep.
next question.
Multi-select. Explain what happens during the formation of maltose. I wonder what that is.
Something probably.
At least something
Nah.
important.
Just a little bit. I think it's D, guys.
Absolutely.
Definitely.
Alright, so the correct answers
Alright.
are A and B.
All right. Okay, so definitely we can rule out D.
Yeah.
That's obvious. Um, but so first thing, remember what dehydration synthesis is and hydrolysis. So hydrolysis is when you take it apart. Dehydration synthesis is when you put it together. Correct me if I'm wrong, because I may be wrong.
Yep, you're right.
Okay.
And C is wrong
My
because
computer's lagging.
that's just another memorization thing. Glucose and fructose does not make maltose. Alright,
Right.
moving on. Question 19. In a phosphorylation cascade, why is the phosphorylation and dephosphorylation of proteins important?
Okay, so make sure you know what phosphorylation and dephosphorylation is.
Yes, that's kind of important. Just a bit.
It's basically the key word of this whole thing. But all right. Does someone want to explain what
Yeah.
dephosphorylation
Is the timer up?
is?
Yeah, now it's
Okay. So the phosphorylation, it is the triangle. It activates and deactivates protein and signal transduction pathways. And what phosphorylation does is it adds a phosphoryl group, which I believe is PO3 negative. And dephosphorylation is the removal of that group. But when a phosphoryl group is added to a protein, it'll cause the protein to undergo a shape change. And it'll also interact with the polar amino acids on the protein. And that will alter the function of the protein, which almost always activates it. And then that protein will go on to trigger other proteins in the pathway and activate them too.
So signal cascade, right? Something like that. Was that the name of it?
Phosphorylation cascade.
yeah.
Oh, phosphorylation cascade. Yeah, it's something cascade. Signal phosphorylation cascade. Whatever the name is. It's yeah.
Yeah.
Oh yeah, it's in the prompt. I
The-
knew.
Yeah.
All right, it's fine. It's fine.
And it's obviously not B because DNA replication is involved with mitosis and interphase and all that fun stuff. And then C and D are about signaling molecules and them interacting with receptor proteins.
Yep. All right, moving on. Question 20.
Ooh.
The pituitary gland sending hormones into the blood to be carried to cells across the body shows what type of signaling?
Yep. Yeah, you don't need to know what pituitary gland is to answer this question.
Yeah.
All you need to know is it's being
just an
carried
example of the
around the body.
signaling.
Yeah, you just need to know it's being carried around the body. So.
Yep.
All right. All right.
Time is up.
All right.
Right, so the answer for this one was C, the yellow circle, endocrine signaling. And endocrine signaling is the main signaling that's long distance, so it'll go across the entire body. And it's not synaptic or paracrine signaling, because both of those are close range signaling, where cells will send signals to their neighbor cells in their general area.
Yeah.
And it's definitely not autocrine signaling, because that cell is sending signals to themselves.
Itself.
Yeah.
Yeah. It's
Synaptic is also just neurons.
Nerves. Yeah. Neurons.
All right. Next
Yeah.
question.
And yeah, if you don't remember, endocrine is hormones as well.
Yep.
So,
Yeah.
or
What are the
endocrine
s-
uses hormones.
What are the three phases
You know what I
of
mean.
interphase, and what highly important event to cell division occurs in interphase?
Ooh.
No, it's not important.
I definitely didn't just talk about interphase a question ago. Nope. That didn't happen.
Nah. All right.
You having fun over there?
All right. Yeah, definitely. So make sure you know your phases, and also on site note, make sure you know your the places where it checks over its stuff. definitely didn't forget the name of it, but it will come to me.
Yeah. So the answer is
What is it called? Hold on.
Okay,
The
while
answer
he's
is
looking
G1 phase, S phase, and G2 phase. The chromosomes/organelles will replicate.
So that's all a portion of interphase. The chromosomal and organelle replication happens in S phase, which is also called synthesis phase. And each of the other options all have portions of mitosis within them. So like prophase, prometaphase, and telophase. So you know that none of those are correct.
Yep. All right. Next question. 22.
Okay. Well,
Explain
never
what
mind.
occurs during anaphase of mitosis. Or now you can explain your thing in a minute.
Oh, okay. Well, I was trying to find cell regulation, but why did we give 60 seconds for this one? All right, anyways. I was thinking about cell cycle regulation. I don't remember what those things are. Oh, checkpoints.
There
Yeah.
we go.
That's what I was talking about. Yeah. G1 checkpoints, etc.
Yeah.
Synthesis, G2 checkpoints.
And specifics.
That regulate.
Yeah, specific stuff
Regulate the cell.
happens at each of the checkpoints to like tell the cell whether or not it should move on with interphase. And we don't have any questions covering that, but those checkpoints and like the different things required for them are detailed in the notes on our website.
Yeah, they're really important to make sure like nothing goes wrong.
Yeah. This next question is not that difficult.
Not really.
You guys probably
I don't know why we gave it
Probably
60
can't get it.
seconds.
I don't know.
Alright, so it's the
Yeah.
cohesion of proteins holding chromatids together are cleaved and they separate.
Yep. So this is knowing your mitosis, meiosis stuff.
Yeah.
And you can tell it's none of the other ones because all of them happen in like different portions of mitosis.
Mm-hmm.
Yeah. Alright.
All right.
Next
So
Alright.
moving on.
23.
All right.
Alright.
What is the
All
purpose
right.
of motor proteins? Why did we give this one
All right.
60 seconds also?
That's
Oh,
I don't know, but that
I
sounded
feel
that sounded like you were about to say something. But it's okay.
I
Say
don't
something
even
after
want to
that,
know.
but it's fine.
Okay.
What do you mean? Okay, all right. There was no implication. It was just the intonation. Oh,
Okay, I think we should just skip the 60 seconds
Yeah,
because we don't need
skip
it.
yeah.
Skip.
All right. So
And the answer
why?
for this one is
Yeah, you can go. It's fine.
what? Okay. The
You can
answer
go,
for
you
that
can
one
go.
was they're the proteins that move chromosomes along microtubules and the anaphase. And that's important because someone's mic just yeeted. The chromosomes need to move to opposite ends of the cell in order for the cell to actually split in half and to make sure that two daughter cells each have the correct amount of chromosomes. So the
Yep.
the motor proteins will either like attached along with the chromosome and pull it up the microtubule, or they'll sit at the ends of the microtubules and like reel the chromosomes in like a fishing pole.
Right. So talking about motor proteins. Oh yeah, and by the way, to add on to Nico or Caitlin, that's weird calling you by your real name, but we're not worried about that. So basically, when it pulls back the chromosomes, it degrades the microtubules. So, or degrades the microtubules. And when we're talking about motor
Yeah.
proteins, I can't not talk about actin. So what actin is, it's basically a motor protein that moves the cell. And how it moves it is by, you know how cells are connected kind of by the extracellular, what's that called? That thing?
Matrix?
Fluid?
I think it's
Question mark?
I think something like that, but it's connected to the system outside of the cell. So the cell's actin basically will like conglomerate near like the end of like a cell, and it would like move along that line, I guess you can say, to wherever it needs to go for resources, materials, that sort. Could you guys should go to our website for resources for for
I love our
bio, you know.
plugins.
Shameless promotion.
You know, great plugins we have. All right, should we move on to the last question? Whoa.
Yay. Which of the following protein pairs aid in the regulation of the cell cycle?
Pears.
Fruits.
Fruits, yeah. All right. Let's see this. Okay.
So it is cyclins and cyclin-dependent kinases.
Yeah.
Right. So I kind of was talking about this, but kind of didn't. CDKs and cyclins. That's, if I'm not wrong, again, that's the checkpoints. But
Yeah, they're involved with the checkpoints.
I'm just afraid of saying something wrong.
Well, I mean, it's kind of in the question, but we
So I
don't worry
want to
about
explain
that.
that.
Yeah.
It's fine.
Mostly the two collectively they're called just the cyclin dependent kinase complex thing. The CDKs that Shadow just said. And their main function is just making sure that all the checkpoints or all like the processes and whatnot is where they need to be for each checkpoint in the cell.
Yep.
And there's there's three main important checkpoints. There's one at the G1 phase, there's one at the G2 phase, and then there's one for like the mitotic phase, which is mitosis, as well as the little end portion of mitosis that I forgot what the name is for, but it starts with a C and it involves the two cells actually completely separating.
Cleavage? Cleaving?
No.
Something like that.
No.
Maybe.
I don't
Wait,
think
hold
that's what it's called.
It's cytokinesis.
I know when it pinches in. I know when it pinches in, it's called cleavage. I know that.
Alright,
Yes,
Shadow.
it is.
There's no implication behind it. It's
Alright,
it's
okay.
literally what it's called.
I know.
Wait, hold on. What? Let me find what the splitting is called.
Alright.
Cytokinesis?
Yeah,
Is it that?
cytokinesis.
I think it's cytokinesis.
It is cytokinesis.
Yeah. It is.
So it's definitely not motor
Okay,
scaffolding
yeah, just
proteins
making sure.
because we just talked about motor proteins before. They definitely do not regulate the cell cycle. Receptor tyrosine kinase and ion channel receptors. Well, it's definitely not ion channel receptors. They kind of are they involved in this function?
No, they're not.
Yeah, I
The
think
receptor,
they are.
the receptor tyrosine kinases and the ion channel receptors are both receptors that are involved in the signal transduction pathways. So both of them will be like hanging out on the edges of the cell to bring in ligands and stuff. So they're not involved in mitosis at all, really. And the same thing with the G protein coupled receptors and G proteins. Those are also involved in signal transduction and not in mitosis or interphase.
Yep. All right. So that's the end. You guys said that we wouldn't have time for an FRQ. I mean, technically we do, but we don't worry about that. We can cover FRQs in the SAT. Not SAT. Did I say SAT? And the AP prep later in the year. Yeah, definitely. SAT, yes, definitely. SAT. All right. So do we do the outro now? Or
I guess so.
That's great. So thank you everybody so much for watching. Make sure you like and subscribe since you guys are on YouTube. Go check out our other resources and notes that are on our website. So we have a lot of different subjects, different AP subjects that you guys can look at, view, or watch. Depends if you're watching the YouTube or Spotify stuff that we did. I am completely failing at this outro, by the way, but
okay.
We don't worry
Just
about
keep
it. Going. Share to all your friends. Thank you guys for supporting us, and
good luck on finals.
Good luck
midterms.
On finals. Finals, midterms, same thing. We'll see you guys next time.
See ya.
Bye.
Our stream has been ended.
Are you